All right, hello everybody, and welcome back. So we're going to start in on adaptive immunity. All right, so what do we mean by adaptive immunity? Okay, so first of all, we're still defending our body against these invading agents, so bacteria, toxins, viruses, cat dander, anything like that. It's going to be different than what we had talked about previously, so it's different than the non-specific defense mechanisms in that it has what's known as specificity. That's actually the biological term for it. So what that means is that it recognizes you or self, right? Recognizes yourself, and it also recognizes other or non-self. Okay, so it's very specific. And it also has a memory, which basically means that the second time that you are infected with something, your immune system is even in higher swing to get rid of it. That's basically how different what's it called vaccines work. Basically, like you get a vaccine, you produce antibodies to whatever the virus is, and then the next time that you're exposed to it, your immune system's already primed. So you have in that second encounter, it's a very vigorous response. So sometimes when you're infected a second time with something, or after getting a vaccine, the response is not nearly as bad. You may not even notice that you've been infected, right? So, I don't know, clear case is something like chickenpox, right? You have it once, pretty much that's it. After that, you can be around people who have the active chickenpox because it's not going to spread because you're already immune to it. Okay. All right. So there's two main types of adaptive immunity. There's cell-mediated and antibody-mediated. So here, let's kind of I'll just take you quickly through this image, and then we're going to talk about each one. So here we have our red bone marrow. Remember, that is where the different lymphocytes are created or are born. So we have the B cells, which mature right in the bone marrow. You can see that arrow going this way. And then the T cells are going to go to the thymus where they mature. As far as the B cells go, you can see them right here. They have these antigen receptors. So that term antigen should be familiar to you. Right, when we talked about red blood cells and like blood types, remember we had the A antigen on type A blood, B antigen on type B blood, or AB or nothing, and then we had the RH. Those are all the antigens on the surface of the red blood cell itself. So similarly, we're going to have these membrane-bound proteins on the surface of these cells, and these are antigen receptors. So they're going to be able to bind to other antigens on the surface of other cells, okay? And we'll talk about why that's important in a second. All right, so anyway, these B cells, at any given point in time, if you're not exposed to anything, they're in their rest stage. But after your immune system is made aware of some sort of microbe invader, they will activate. These B cells basically will start dividing. They turn into the active form, which is the plasma cell, and then the plasma cells produce tons and tons of antibodies. Okay. At any given point in time, when these B cells divide, not all of them will be turned on. Not all of them will automatically go into that active phase. Some of them remain dormant, and those are called memory B cells. Okay. So let's just look over here in the thymus area. Right, pre-T cells come from the bone marrow to the thymus. There they mature, and they can turn into one of two types of T cells. We have what are known as cytotoxic T cells and helper T cells. And one of the things you'll notice here is these cytotoxic T cells have something called a CD8 protein. You see these two little things sticking out of it. Whereas the helper T cell only has one, so it's called a CD4 because half of 8 is 4. Right? Kind of can hopefully see that in the image. Let's just take a quick look at the cytotoxic T cells. Again, both of these types are dormant unless there is an invader. Once they are activated, they are going to divide, and you have the active form and then as well as a memory form. Again, and that memory form is going to be important for that second time you're exposed. Okay, kind of like you already have a reserve army waiting, for another invasion or something. Okay, and you'll see here that cytotoxic T cells, they are going to actively leave the lymphatic tissue and attack invading antigens. Okay, and you can kind of see this little shorthand here. Cell-mediated. This consists of cell-mediated immunity, which means it's directed against intracellular pathogens, cancer cells, and tissue transplants. So the cell-mediated immunity means this cytotoxic T cell is going to go and kill another cell. Okay, again, in these, well, we'll get there in a second. The helper Ts, you'll notice they're not going anywhere and attacking anything. They are doing just that. They're going to help. Again, they'll be dormant unless something comes along. Once they activate, they will divide. Once again, we'll have the active form as well as the memory form of that. And the active T cells are going to help and stimulate the cytotoxic Ts and then the plasma cells so that, again, we have this really robust response. Okay. So that's kind of a quick overview. Let's get a little bit more background information, talk a little bit more specifics. We have something called a major histocompatibility complex antigen. Okay, so again, we're talking about antigens. So these are going to be these proteins on the surface of cell membranes. Okay, there are two types. There's something called an MHC1, and these are molecules that are built into the cell membrane of all cells except red blood cells. Okay, so all cells have them except red blood cells. They do not. They have their own special antigens, and we already talked about that. And then we have MHC2s. These molecules appear on the surface of antigen-presenting cells only. Okay, so MHC1, you will find that in every cell in your entire body will have these MHC1s, all of them have this, except the only one exception in your entire body is a red blood cell. So basically you go, does my epithelial cell have an MHC1? Yes. Does my kidney cell have an MHC1? Yes. Does my eyeball cells have an MHC1? Yes. Red blood cell? No. Okay, so everything except for red blood cells. And then MHC2, these are going to be a slightly different type of antigen, and again, they only on the surface of antigen-presenting cells. That's it. Okay. All these MHCs, they're transmembrane, so they go across the membrane. Glycoproteins, meaning they're part sugar, part protein, and they are self-antigens. That means unless you have an identical twin, your MHC antigens are unique. They are one of the markers that say that you're, whoever you are. So I'm Liz. All my cells have my special little MHC on them that is unique. It's like your fingerprint, I guess. Okay. Again, remember we just mentioned just a slide ago that those cytotoxic Ts will kill cancer cells. It's these MHC1s that they will recognize. So like if you got a transplanted tissue from somebody else, those cytotoxic Ts go around, they go, whoa, whoa, whoa. Whose kidney? This is not my kidney. It does not have my name on it. It is not my fingerprint. This is just one giant, to them, it's one giant, big invading microbe. They can't, they don't know. It's not like they have brains. There's no way for them to know that you actually need this kidney, otherwise the whole body dies. That's not how they work. All they know is that it's not you. So we have to get rid of that thing that is not you, despite the fact that you might actually need it to survive. And so they will actually attack it. So that's the reason why transplanted tissue becomes rejected. Okay. So let's talk about this MHC1 first. All right. So basically, remember, MHC1s, we're going to find this on the surface of all cells except red blood cells, right? So the reason for this, all right, and I don't know, maybe reason for this is a little bit too strong, but what this means is that if a foreign antigen is present inside body cells, so a foreign antigen would only be present inside one of the cells of your body if some microbe, bacteria, virus, something got inside of you. Okay, so something got inside of one of your cells, your body will present it. Okay, most cells can process and present endogenous antigens. Okay, so basically, and if it's within you, okay, if it's within the cell can go, no, this thing is inside of me, right? It'll actually kind of raise a red flag, if you will. It'll show that, hey, it's not actually me. Instead of it saying that's me, it says there's something else inside of me, and that's the cell. Okay, so again, things that will get inside are viruses. Again, bacteria sometimes can get in there, and they're producing these toxins, and they can be presented on these MHC1s or abnormal proteins produced by cancer cells. So let's take a visual look of this, what this actually means. Okay, so here's a cell. I don't know, pick one, like an epithelial cell, right? Remember we did all those in 201? So let's pick a nice little epithelial. Well, it's it's kind of like a simple cuboidal epithelial cell inside the tubule of your kidney. Okay? Now it's just, that cell was just sitting there minding its own business, and all this little green MHC1 self-antigen was presenting a nice little peptide fragment, antigen fragment, saying, hey, this is me. This cell belongs to, I don't know, in my case, Liz, right? Well, somehow something got inside, or maybe it started turning cancers, something. Okay, the cell is going to break up that antigen. It will put it into a vesicle. Okay, remember when we talked in 201 about the endoplasmic reticulum and how those proteins then get sent to the Golgi apparatus, they get packaged into vesicles and presented and then sometimes inserted into the cell membrane. Okay, that's what we're talking about here, that process. So basically, again, you're going to break down this endogenous antigen. If it's a foreign one, you break it down. You package it into that vesicle. It will bind to that MHC1 that your body has produced. Again, you pack it into a vesicle, and it inserts itself into the cell membrane. So now when one of these cytotoxic T cells comes around, it goes, whoa, 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 something's not right with this cell. Okay, that's that red flag that there's an invader. Right, hopefully that makes sense to you. So your cells basically have the ability to alert the immune system to say, something's inside of me. We have foreign invaders. Okay. Now MHC2, remember, we're going to find MHC2s on the surface of antigen-presenting cells only. Okay, so it's a little bit different than the MHC1. Okay. These antigen-presenting cells are special class of cells. So these are examples of them are dendritic cells, macrophages, and B cells. That's it. Okay, just those three. So dendritic cells, macrophages, B cells, those are the ones that will have MHC2. No other cell does. And basically where we find a lot of these, we'll find a lot of these antigen-presenting cells, like in the epidermis, dermis, mucous membranes, respiratory tract, gastrointestinal, urinary, reproductive tracts, right? Anywhere where we might come into contact with some foreign microbe. Okay, lymph nodes, right? We saw that. Those particularly the dendritic cells were in close proximity to cytotoxic T cells. Okay. So what it's going to pick up on are the antigens that are present in the fluids. So just a second ago, with the MHC1, we didn't have to worry about things that were in the fluid in that case because it already invaded the cell. It already left the fluid and it actually went into the cell. So these, again, the MHC2s that are located on the antigen-presenting cells, these guys are going to be patrolling the body for the things that are outside of cells that haven't infected them yet. So bacteria that haven't infected a cell yet. Bacterial toxins, parasitic worms, for example. Okay, pollen, dust, viruses, again, that haven't infected a body cell yet. So let's just take a look at this process. So in this case, we have an antigen-presenting cell, right? So this big blue blob here is the antigen-presenting cell. So you pick whichever one you like, monocyte, dendritic cell, B cell. And on the surface of its cell membrane is the MHC2 self-antigen. And again, normally it would have these peptide fragments that say it's you, it's Liz, and that's who it is. Now, what's going to happen is that this macrophage or dendritic cell is going to pick up on that chemical cue of that antigen, of that virus, that bacteria, whatever, and then it's going to gobble it up. It's going to take it into itself because again, it's not in the cell yet. So it actually has to phagocytize that antigen. Okay, it then has to break it up, and again, it's the cell. It's a regular cell, so it has that rough endoplasmic reticulum. It has the Golgi apparatus, so it's producing that MHC2 in the normal standard cell fashion, right? It'll go through transcription, translation. Ribosomes in the rough ER are making this. It gets packaged into vesicles by the Golgi apparatus. So basically, these two vesicles, both the one that's containing the broken down fragments of the antigen, as well as the vesicle that's containing the MHC2, are going to combine. The antigen fragment as well as the MHC2 come together, and then basically that's inserted into the cell membrane of the cell. So now that dendritic cell, that monocyte, that B cell, has this, and it can go and present it. It can go and show it because now it's on the outside. So now when a T cell comes up to it, it's like, whoa, what do you got on? That's not you. I don't know. It would be sort of like you coming home with someone else's shirt on, and your significant other's like, whoa, whoa, whoa. That's not your shirt. Whose shirt is that? Right? Something's going on. It's that kind of response. Not a good one. Right? Well, maybe in your case, maybe in the case of the immune system, it is a good one because you want it to fight off the invader. I don't know if you want your significant other fighting off the other person, but kind of that analogy works. So we'll go with that. All right. So hopefully this makes sense to you. We have MHC1s and MHC2. Okay. So why do we need them? So for an immune response to occur, those B cells and T cells have to recognize that there's a foreign antigen present, right? I mean, you can't do something about something that you don't know exists, right? Now, B cells can recognize, remember, B cells are staying put in those lymphatic nodules or in those lymph nodes. So B cells can recognize antigens in the lymph, in the interstitial fluid, or blood plasma. That's about it, okay? T cells can only recognize fragments of antigenic proteins that are processed and presented a certain way. So in other words, B cells are sort of sitting there as long as that interstitial, well, not the interstitial fluid, the lymph brings the antigen to them, they can process it themselves and go ahead and present it. Okay, B cells are a little bit more self-sufficient. T cells can only recognize fragments of the antigenic protein. A T cell will literally allow a full-blown bacteria, virus, whatever, to go right by it. It can't process that. It just can't process it. It has to have it broken down and presented to it in a very specific way. Okay. And again, those antigen presenting cells, they'll pick up on whatever it is, and as we saw in the lymph nodes, they usually are very close to the T cell, so things don't just go right by the T cells. It'll, be picked up by the dendritic cell, which will then present it to the T cell right next to it. But in case, let's say, the antigen presenting cell finds it somewhere farther away from a T cell, the antigen presenting cell will go toward the lymphatic tissue, where they will then present the antigen to T cells. Okay. And not all T cells and not all B cells will be able to respond to all different types of viruses and bacteria and stuff. You're going to have that specific shape that it can recognize. Okay, this is why you're not immune to everything. You have to have that specific, shape, I guess, I don't know how to describe that, for it to recognize it. Okay, so it is a possibility that your dendritic cell or your antigen presenting cell is trying to present something to a T cell, and T cell's like, yeah, I still do, and not one T cell can recognize it. Okay, so not one will respond to it. And I don't know if you can hear that. I apologize that those are my neighbor's barky dogs. All right, I'm actually going to stop this right here. After this, we'll get into cell-mediated immunity, and then after that, we'll get into antibody-mediated immunity. All right, I will see you guys next time.